Please rise. Order! We will have order in my courtroom. You there! Give me back that pen. Sit down. Alright, so, the lawyer for the plaintiff, you can examine the witness if you want.
Ms. Klein, can you explain to me how genomic instability is caused by cancer?
Almost every type of cancer is characterized by changes in the genes of the cancer cells when compared to normal cells. These changes are usually referred to as genomic instability. It is also well known that factors within the environment are an established cause of genomic instability.
It's very interesting, Ms. Klein. Is there any evidence that suggests that smoking is the cause of genomic instability?
75% of lung cancers seem to be related to smoking cigarettes. It is important to realize that cancers in non-smokers demonstrate clear areas of genomic instability. However, when the cancers of smokers are studied, they do exhibit areas of genomic instability that are different than those seen in non-smokers.
Ms. Klein, in your opinion, is it more likely than not that smoking is the cause of genomic instability found in smoking-related lung cancer?
Yes, it is.
No further questions, Your Honor.
Are we done here? No? Oh, okay. Well, the defendant, you can cross-examine if you feel like it.
Miss Klein, you have stated that environmental factors can cause genomic instability. I don't see the association.
Normal cells have complex mechanisms in place to repair damage to DNA. Almost all cancer cells have abnormal genetic material. It has been shown that changes in specific genes reduce the ability of cells to repair their own DNA. When there are errors in replicating DNA which cells cannot repair, this leads to mutations which can lead to formation of cancer. Cigarette smoke contains large numbers of chemicals that can bind to DNA and disrupt the replication process.
That sounds abstract. Can you give me specifics?
There is an example of a cell line derived from a small cell lung cancer, which has been shown to have a large number of mutations that are linked to chemicals found in cigarette smoke. This process begins when the chemicals in cigarette smoke bind to DNA. This results in a process known as base transversion mutation, where pyrimidines are switched to purines and vice versa. This disrupts the information stored in the genetic material.
Do you have any proof?
The research done by Thu et al. And Cassidy et al. Clearly proved this is the case.
Oh, witness, you can leave the evidence here. I'm not quite done my drawing yet.
Could you explain to us why an increased metabolism is necessary for cancer cells?
Cancer cells need to make adjustments of metabolism in order to fuel cell growth and division. As a growing tumor with increased metabolic demand decreases its nutrient supply, it needs a way to maintain this. If not, the cells will undergo apoptosis.
What is the role that increased cell metabolism plays in cancer?
Cancer cells program their metabolism and energy production by limiting the majority of it to glycolysis. The majority of pyruvate generated is converted to lactate and lactate dehydrogenase. This conversion allows lactate dehydrogenase to recover the NAD+ needed to maintain glycolysis, which is critical for the maintenance of growth.
It was said earlier that cancer is induced by genomic instability. But wouldn't the body fight that off?
Normally, the body is expected to initiate programmed cell death in response to stress by utilizing Fas ligand acting in the intrinsic pathway, leading to a cascade of proteolysis and eventual cell death. However, the BCL-XL, BCLW, MCL1 proteins can act as anti-apoptotic and stop the process. Under the prolonged periods of stress, the proteins and ligands fail to induce the apoptotic process in cancer cells. This inhibition of cancer cells is known to be caused by carcinogens.
Please get to the point! So far, you have only illustrated the program of cell death. You have not offered any arguments against me.
Even though the conditions that trigger apoptosis are not entirely understood, several receptors have been identified in triggering apoptosis. Most notable is a respected member of the tumor suppressor, the p53. The TP53 gene induces apoptosis by upregulating expression of NOXA and PUMA BH3-only proteins, doing so in response to substantial levels of DNA breaks and other chromosomal abnormalities. The death of the TP53 gene activation severely affects the damaged sensors, which essentially destroy the apoptosis-inducing circuitry. The tumor is now infested with only anti-apoptotic regulators, BCL2 and BCL-XL, which allow the tumor to evade cell death and grow continuously.
You are simply speculating. You have no proof that carcinogens killed cell death program.
This is not true. Adam and Corey, in their revolutionary research of 2007, proved that PUMA is a direct transcription target of p53, and that these pathways might install some sensitivity. However, the elimination of p53 prevents such a cascade, and now the program has been eliminated.
Mr. Mazumdar, please explain what metastasis is and how cancer cells invade the body.
Metastasis refers to the formation of cancer cell colonies throughout the body. So obviously cancer cells have to invade for this to occur. It has been shown that cancer cells can invade and metastasize when cadherin and catenins lose their ability to allow normal cells to stick together.
I see, but what does this have to do with smoking?
Well, in one survey, 148 patients were surveyed and 93% were either smokers or ex-smokers, so it is very likely that smoking was the cause of cancer.
Would you agree that smoking can cause invasion and metastasis of cancer cells?
I would.
Ilyas, tell me about telomeres.
Telomeres are made from tandem repeats of 6 nucleotides at the ends of chromosomes with a 3' overhang.
Overhang? What?
Well, it loops around in a circle and gets stabilized by a protein that's called telomeric repeat binding factor 2. Without this cap, chromosomal fusions would not be preventable. What is happening is that when telomeres become critically short, p53, a tumor suppressor protein, causes cell cycle arrest, and so cells stop dividing. This occurs to prevent genomic instability and acts as one of the cancer brakes.
Order! Order! We will have order in my courtroom. So, according to the undeniable fact that smoking is very bad, and in light of the new evidence provided by the plaintiff, the court decrees that the defendant, Zaruch Beg, yes, guilty and is responsible for damages incurred by smoking.
What?
So I thereby sentence you, Zaruch Beg, to utilize a nicotine patch, maybe gum, whatever you want to do, and quit smoking. I'm telling your mother. Order! We will have order in my room. Sorry, do it again.
Telomerase is a ribonucleoprotein that elongates the 5' end of
Oh, that's it. Sorry.
Ms. Klein, is it more likely than not?
Do you have any proof?
Ms. Klein, is it in...
So, do you swear to tell?
It acts to prevent cell cycle arrest and even apoptosis because it doesn't signal. It doesn't. Sorry.
Objection, Your Honor.